for being here. Great. It's a real pleasure to welcome John Bickle to the Rotman Institute and the Brain and Mind Institute here at Western. So I've known John for a very long time.
Exactly.
I probably shouldn't have had to write anything out, but I always like to do that. So John Bickle is professor of philosophy and a faculty member in the Shackles Honors College at Mississippi State University.
Yep. Mm-hmm.
His work in philosophy of neuroscience historically is focused on a broad range of topics, including mind-brain reduction, what John himself dubbed the strategy of reduction in practice in cellular and molecular neuroscience. And he's also focused on experimentation and most recently tool development in neuroscience. Yep. And throughout his career, John has had his finger on the pulse of neuroscience, actively engaging in collaborative projects with neuroscientists to push forward philosophical and scientific thinking about the science. And his work has appeared in top-tier journals in philosophy, including Philosophy and Phenomenological Research, Philosophy of Science, Biology and Philosophy, and Synthèse. And he's author of Psychoneural Reduction, The New Wave, in 1996 by MIT, Philosophy of Neuroscience, A Ruthlessly Reductive Account, and it turns out he's still ruthless, maybe less reductive, but he's still ruthless. And with co-authors Alcino Silva and Tony Landreth, he worked on Engineering the Next Revolution in Neuroscience, The New Science of Experiment Planning, which was published by Oxford in 2013. He's also editor of the Oxford Handbook in Philosophy of Neuroscience and co-editor of a recent book, The Tools of Neuroscientific Experiment, Philosophical and Scientific Perspectives. So one thing I'd like to say about John, I've known John for many years. I've known him since I was a graduate student myself. And he's been instrumental in building and shaping the field. And he does this by encouraging and mentoring budding philosophers of neuroscience from all over the world. And he also has created a number of exciting and stimulating venues for them to present their work and network with their peers. In fact, he founded and directs the Deep South Philosophy and Neuroscience Work Group, which organizes an annual conference every year, bringing together established and up-and-coming neuroscientists and philosophers to talk about their work and get feedback. Indeed, I stand before you today in part due to John encouraging me many years ago and supporting me throughout my career. So without further ado, please join me in welcoming John, who will be talking to us about unnoticed features of exploratory experiments revealed in the 60-year history of the NMDA receptor.
Great, good. Thanks, Jackie. Thanks so much. Thanks, everybody. Wow, what a great visit. I just had an awesome time and great discussions, got to see all kinds of the cool work going on here. Just awesome. And I'm really glad I'm giving this talk here today because, as you'll see, a number of Canadian scientists figure prominently in the story I'm going to tell today. All right, so let me just kind of give you a little bit of background. So as Jackie mentioned, in 2014, Alcino Silva, Tony Landreth, and I published this book, Engineering the Next Revolution in Neuroscience, The New Science of Experiment Planning from Oxford. And basically what we did at the very beginning of that book was kind of cull out of the literature the 25 to 50 most cited papers from the field of molecular and cellular cognition. And from that collection, we derived the different types of experiments that are done in the most prominent work in this field. And we distinguished what we called identity experiments, tool development experiments, and connection experiments. Connection experiments are the kinds of experimentation by which you try to establish a causal connection between one neurobiological kind and another. And because those kinds of experiments are so prominent in molecular and cellular cognition, we spent the entire 180 pages of the book explicating that kind of practice and how those experiments are done and how their results are integrated across labs, across experiments. And so that left these other two kinds of experiments relatively unexplicated in the book. Now, over the last half decade, I've spent a lot of time figuring out how the tool development experiments in molecular and cellular cognition work and how those experiments go. But with regard to identity experiments, here's where the status of that concept stands. Early in the book, we say our framework sorts experiments in molecular cellular cognition according to their fundamental goals. 1, attempts to discover new phenomena and understand their properties, identity experiments. And then we give a couple of examples of identity experiments in an early chapter, but since then, Nothing. None of us have said anything about the way that these kinds of experiments work, the kinds of experimental practices by which you engage in identity experiments. Well, no time like the present to flesh out this skeleton. So what I want to do is I want to take a first meta-scientific exploration of identity experiments in neurobiology, and I want to focus on the discovery of the N-methyl D-aspartate receptor and its fundamental properties and activities. I think this is a wonderful case to explicate this notion. Now, spoiler alert. At the end today, I'll argue that some of these Silva, Landreth, and Bickle identity experiments illustrated by this history turn out to be exploratory experiments, what philosophers of science for the last couple of decades have tried to characterize as exploratory experiments. But that literature is still pretty sparse, okay? So I think what I'm going to try to point out today is this history reveals three features of identity experiments that have not yet been noticed in the published literature, and there may be other lessons for the philosophy of science lurking in this wonderful history. Okay, so let me just first give a little background on where we stand right now with regard to N-methyl D-aspartate receptors. First of all, the chemical neurotransmitter that activates these receptors most primarily is glutamate, the most prominent excitatory neurotransmitter, with some estimates now estimating that maybe up to 50% of all mammalian synapses involve glutamate as a neurotransmitter. The full pharmacology of glutamate has now been worked out from its derivation in presynaptic terminals, its binding into vesicles, its release, and its reuptake both directly into the presynaptic cell as glutamate, and its reuptake into glial cells and conversion back into glutamine, and then its synthesis in the cell. So that's all been worked out in very, very detailed neurobiological detail. With regard to the receptor molecules, we now know that there are numerous kinds of glutamate receptors, and both ionotropic and metabotropic glutamate receptors. Now, the N-methyl D-aspartate receptor, and again, I know this is just background for a lot of you, but it's one type of ionotropic glutamate receptor with some remarkable features. These are only activated under conditions of high presynaptic neuron activity because it requires both the glutamate and the glycine to bind to extracellular domains. And the reason for that is because there's a magnesium ion stuck in the middle of that channel. And so even if the channel is open with that magnesium ion stuck in there, no efflux or influx of ions can occur. But that magnesium ion is voltage dependent. When the voltage of the postsynaptic neuron is depolarized enough, generally depolarized more than threshold, that magnesium ion pops out to allow the influx of various kinds of ions, most importantly, calcium. And so because of this nature of the activity, because of the underlying structure of this particular protein, they came very quickly to be called coincident activity detectors, because they allowed for ion influx, especially calcium influx, only with co-occurring high activity in both pre- and postsynaptic neurons. Now, this probably ringing some bells for many of you from the writings of Donald Hebb, the Canadian psychologist. In this passage that's probably been quoted, I would guess, 30,000 times. Here's what Hebb says about Hebb's rule or postulate. Let us assume that the persistence or repetition of a reverberatory activity or trace tends to induce lasting cellular changes that lead to its stability. When an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing B is increased. You've probably heard that line, I don't know how many times. Okay, now, however, when we take the slogan that Hebb's thing, cells that fire together, wire together, and ask the mechanistic question, how might cells that fire together, wire together, in Hebb's wildest dreams, because he died in 1985, and you'll see where we were with understanding these receptors. He could not have imagined a more efficient mechanism than N-methyl D-aspartate receptors to realize the mechanism of his postulate. Okay, and the reason is because that influx of calcium ions, that influx of calcium ions through the activated N-methyl D-aspartate receptor, calcium is now known to be an important second messenger that leads to intercellular activity for the variety of effects, including synaptic plasticity, a form of synaptic plasticity that has been actively tied on the experimental bench to learning and memory. I won't go through the details here. I'm sure some of you know them, but basically the calcium influx is an important second messenger to drive not only the inserting of additional other kinds of glutamate receptors into the postsynaptic density, but it also drives active gene expression, new protein synthesis to actually restructure the postsynaptic density of the postsynaptic cell. Okay. So now more of what we know, NMDA receptor dysfunction has now been confirmed in Alzheimer's disease, other forms of dementia, schizophrenia, depression, ischemic injury post-stroke, and more I'm sure will be revealed. And many of you may know of this particular pharmaceutical, Namenda. Namenda is an N-methyl D-aspartate antagonist, and it is a cognitive enhancing medication. It can treat dementia associated with moderate to severe Alzheimer's disease, but do know that it's a very modest effect. Okay. Furthermore, in just a decade ago, we now have the structure and function relations have only recently been confirmed at the atomic level by X-ray crystallography and electron microscopy. But here's the thing. It was a long journey to today. 6 decades of what Silverland and me call identity experiments. I mean, it goes back to the time of the classic 58 Ford Thunderbird. Okay, so today I'm only going to have time to scratch the surface of this history. And I'm going to be telling the episodes that I think best reflect the philosophical lessons I want to draw. But you may see other lessons in this, and I want to hear what you think might be some of the lessons from this remarkable history. Okay, oops, sorry, oops, oh, what did I do? There we go. Okay, so let's start in Canberra, Australia, in the late 1950s, in the lab of not even yet Sir John Eccles. Okay, now, for all of you who are computational neuroscience, you might also know that one of Eccles' lab partners was Wilfred Rahl, who, of course, discovered the cable theory of membrane potential. Now, having successfully demonstrated the chemical nature of neurotransmission, for which he was later awarded a share of the '63 Nobel Prize and his Knight Bachelor, Eccles and colleagues were isolating and identifying potential chemical neurotransmitters. And that discovery of Eccles is worth a talk in and of itself. And I'll be happy to tell you some of the details of how Eccles came to discover the chemical nature of neurotransmission. It's a wonderful history, and it involves philosophy as well. But I'll set that aside for the discussion, or maybe beers later. Okay, so now, why start examining glutamate as a neurotransmitter? What was the motive to start even looking at that particular small molecule amino acid as a neurotransmitter? Well, here's what happens. Jeff C. Watkins, a young chemist, Australian native, trained at Yale, returned to Australia to work in Eccles' lab. So he came back from the States, back to Australia, to work in Eccles' lab. And here's a reflection that he introduced in a publication where he was reflecting on his role in these N-methyl D-aspartate receptor discovery. And he talks about his early collaborations in the late 1950s with physiologist David Curtis in Eccles' lab. Here's what he said. I drew up a list of the many chemical constituents of the brain that had already been reported. Sodium glutamate was at the top of that list because of the high concentration of glutamic acid in the brain. Oh, and because we had a 500-gram bottle that's sitting on the shelf. Now, we did not know of Hayashi's work at the time, which there's no reason they would have known. It was published in this obscure Japanese journal. It was only rediscovered decades later. Now, here's Watkins talking. As a chemist, my interest lay in investigating structure-activity relations of glutamate analogs, the non-selectivity of the action of glutamate. Now, I'll stop here because some of you may not be all up on your biochem. Okay, but the non-stereoselectivity of glutamate basically refers to the fact, like all amino acids, glutamate occurs in two isoforms. The D isoform and the L isoform, depending on the orientation of the amino group relative to the central carbon. Now, it's the L isoform that in most amino acids is the naturally occurring amino acid. But here was the weird thing about their initial explorations of glutamate. The L and the D form, they couldn't distinguish between the two at these receptor sites that were activated by glutamate. And so as a chemist, what Watkins knew what to do next, that demanded the study of more bulky substituents where stereoselectivity might be more apparent. They were just looking around, okay? And they had a chemist here who said, okay, what's the next most bulky analog of L-glutamate? Well, guess what it is? N-methyl aspartate. And that's why they started introducing this as a potential antagonist, as a potential agonist or antagonist of these glutamate sites. Oh, two more to call attention to that you're going to learn about later. Quizquilate and kainate. And so we'll talk more about those bulkier substituents in just a minute. Okay, so that's why N-methyl aspartate, both in its L and its D forms, were introduced into this exploration of glutamate. It was all on the behest of a chemist who realized that the non-stereoselectivity of glutamate at these sites was weird. And so he was looking for something that was a bulkier analog that might show stereoselectivity. Now, glutamate, on the basis of that discovery, that the N-methyl aspartate had different activity when you introduce the D isoform and the L isoform, on the basis of their remarkable studies of these different agonists and antagonists of the glutamate receptor. As early as 1960 and more substantially in 1965, Curtis and Watkins proposed a three-point model for the structure of glutamate receptors to bind charged components of glutamate and its chemical analogs. So just simply on the basis of what activities were done, they proposed that the model had this three-part structure. The receptor had this three-part structure, though of course no one knew anything at all about the structure of that protein at the amino acid level. Now, the mid-1960s to the early 1970s, the neurophysiologists dominated the exploration of these glutamate receptors. Numerous labs, but first and foremost, Hugh McLennan's at University of British Columbia, reported differing responses to glutamate and these analogs in the rat thalamus and in the rat spinal cord. And by the mid-1970s, the evidence was so prominent that the idea of distinct types of glutamate receptors came to be widely accepted, of course, within this tiny little field of researchers. Okay, next insight. This can only be described as sheer serendipity. Okay, so here's the setup. Watkins leaves Australia and takes a job in the medical school at the University of Bristol in the UK in 1973, where he immediately teams up with Biscoe, a physiologist, and Evans, a pharmacologist, and they begin testing, quote, new substances as possible antagonists of amino acid-induced excitation to check for selective antagonism of one excitatory amino acid relative to one another. They were just exploring. They were just trying to test all these different kinds of substances and to see exactly what they would get. Now, here's what Watkins says about their first important discovery. Our first success in differentiating excitatory amino acid-induced responses from one another was, unexpectedly, the mere inclusion of magnesium ions in the medium superfusing the isolated frog spinal cord, which we had adopted for our screening studies. So basically, the reason why they adopted the frog spinal cord is because that's what Watkins had been using back in Eccles' lab. And he was totally familiar with it. It was a very common preparation, and so that's what they were going to use because it was already known that the frog spinal cord had response sites that differed to some of these different excitatory amino acids. So that was their choice because of convenience and because of the fact that so much had already been done with that particular preparation. But here's the thing. Why introduce magnesium? Hey, here you go. Frog ringer solution is traditionally magnesium-free, and we initially introduced it to try to abolish synaptic release of transmitter that occurs at the neuromuscular junction. They were just looking for something they could infuse into the frog ringer solution, positively charged ions that would immediately halt neurotransmitter release. What was the first one, most obvious candidate? Well, there's no magnesium ions in frog ringer solution, so let's just throw that in there. To our surprise, remarkably low concentrations of magnesium greatly reduce synaptic activity, but crucially, only responses to some amino acids, but not others. NMDA-induced responses were almost abolished, while responses to kainate and quizcalate were almost unaffected, and response to L-glutamate was reduced to an intermediate degree. And so, the role of magnesium discovered now, of course, they had no idea how this was happening, right? You didn't even know the structure of that protein at this time. But that's how magnesium got into the study of these glutamate receptors. So by 1977, Davis and Watkins are ready to produce an initial classification of glutamate postsynaptic responses. Quote, we initially classified depolarizations, not receptors, depolarizations produced by glutamate analogs as magnesium sensitive and magnesium insensitive responses. Okay, well, 73 to 81 was a time in which the search was on for new and specific glutamate antagonists. And in this period of time, we get the first classification of distinct glutamate receptors became mentioned. Now, the discovery of numerous substances that produced an, quote, almost identical pattern of antagonism of excitatory amino acid-induced response to that produced by magnesium ions, they resulted in an increasing selective set of antagonists for magnesium-dependent responses. Now, this was a search that was led by biochemists, not neurophysiologists. Here's an example of one of the key papers that was published in this time frame. 2-APV, a potent and selective antagonist of amino acid-induced and synaptic excitation, where they describe the derivation of this particular substance and the results of applying it to these physiological preparations. Now, what led this group to derive 2-APV and test it neurophysiologically for its antagonistic effects on magnesium-dependent responses? The answer to that question? Basic organic chemistry. 2-APV is the next compound in the analog series, meaning compounds that are arrangeable in a linear sequence, all of which share a similar chemical structure, but differ with respect to one particular component. It was the next in the series after D-alpha AA, which had already been discovered, already been derived, and already been shown to be an antagonist of magnesium-dependent responses. Also, 2-APV is the next higher homolog. Compounds that share a distinct functional group, but different by a single repeating unit with different numbers that unit to form a linear sequence after 2-APB, which had already been derived and already been shown to be an effective magnesium-dependent response antagonist. So to a chemist, this was the next structure. This was the next choice of structures to derive and test. And it turned out to be a far more potent magnesium-dependent response antagonist than either of its two from which it was derived. And let me just show you one more thing about this paper I kind of skimmed over. The authors, John Davies and good old Jeff Watkins. So Watkins was the lab at Bristol, derived the compound. They were the ones that chose that compound, and then Davies led the team that tested it for its antagonistic effects. And you'll see that name again soon. Okay, so by the early 1980s, we certainly did not have our current classification of excitatory amino acid receptors, okay? But we were getting there. We were on the path there. And the term NMDA receptor to replace magnesium-dependent response first appears in Watkins and Evans' Annual Review of Pharmacology and Toxicology 81 paper, now been cited over 2,300 times. So now the term N-methyl D-aspartate receptor is in vogue. All right, now the next major breakthrough involves, I mentioned the huge later import of 2-APV. The next major breakthrough comes at the hands of people studying the newly discovered phenomenon of long-term potentiation. Because next came the work of another Canadian, Graham Collingridge. Well, he was Brit, but he's now been in Canada for years. A trained pharmacologist, Graham Collingridge. Okay, now Collingridge has been described as well situated for doing this next piece of research. He studied chemistry as an undergraduate pharmacology student at University of Bristol with guess who? Jeff Watkins. Okay? He earned his PhD in pharmacology at University College London under the direction of, guess who? John Davies. And in the early 80s, he was postdocing at the University of British Columbia under the direction of Hugh McClellan. Isn't that awesome? Like, there's the guy that's ready to go. Okay, now here's what Collingridge did. He used a chemistry research tool to investigate neurotransmitters and postsynaptic responses involved in the Schaefer collateral CA1 pyramidal synapses in which LTP was induced. Now, this was a tool that was brand new to neuroscience. The very first Trends in Neuroscience review paper of the use of this tool in neuroscience had only been published in 1980, and Collingridge was already using it in McClellan's lab. But a tool that chemists like Watkins and pharmacologists like Davies have been using for years. Basically, it's the delivery of minute amounts of substances directly onto specific cell components by ionophoresis, the release of substances from micropipettes by tiny electric currents. Okay, so Collingridge is able to show that the selective NMDA antagonist, APV, applied to dosage that abolished responses to N-methyl aspartate had no effect on excitatory postsynaptic potentials, but it reversibly prevented long-term potentiation evoked by high-frequency stimulation of the Schaefer collateral commissural pathway. So you dump tiny amounts of APV onto the dendrites that you're directly measuring your excitatory postsynaptic potentials from. You give the stimulus that induces long-term potentiation, and you get none. You wait time for that to diffuse. You introduce your stimulus that produces long-term potentiation, and there goes your long-term potentiation. Cool. Okay. So here's what he concludes. Although NMA receptors do not appear to be involved in neuronal synaptic transmission, they may play a role in synaptic plasticity. This was a huge step forward to what we now know as one cellular mechanism involving these N-methyl D-aspartate receptors of LTP involving N-methyl D-aspartate activation and the distinct roles played by these different types of ionotropic glutamate receptors. This was a huge step forward in what has become our current model. Okay, now over the next 3 years, neurophysiologists took over the study. Gary Lynch's lab discovers the necessity of postsynaptic calcium influx to induce LTP. Philip Asher's lab, using the then brand new patch clamp, discovers that magnesium ions actually block the channel. under resting, hyperpolarized, and even slightly depolarized membranes. So we get an idea of how these magnesium ions are blocking the blocking activity in these receptors. And in '86, Amy McDermott, working in Mark Meyer and Gary Wisbrook's lab, demonstrates that the calcium influx via LTP is exclusively through activated N-methyl D-aspartate receptors using the then new calcium indicator dyes, which had just been introduced. Okay, let's go to 1986 now, because here we're going to get the impact of a behavioral neuroscientist on the discoveries about the neurophysiology of N-methyl D-aspartate receptors, and it's good old Richard Morris was posing in front of his famous water maze. Here is the paper published in Letters to Nature in '86, Selective Impairment of Learning and Blockade of Long-Term Potentiation by an N-methyl D-aspartate receptor antagonist, AP5. Now, that paper's been cited over 4,000 times. But notice two of its authors, Gary Lynch and Michelle Baudry. Here's what Lynch tells me in a private conversation. Richard spent a few months in my lab to learn the secrets of LTP, during which the three of us, Baudry, Morris, and myself, dreamed up the idea of trying to show that NMDARs are critical to learning as well as to LTP. The paper turned out, to my absolute surprise, to be immensely influential. The results seem obvious. Gary Lynch. Okay, in any event, Richard gets the lion's share of credit for converting the physiology into behavior. Now here's basically what Lynch did, just in a nutshell. Okay, he did the chronic intraventricular infusion of the N-methyl D-aspartate receptor antagonist DL2 amino DLAP5, also known as 2-APV. He used the very same chemical that Collingridge had used, and Collingridge used the very same chemical that Watkins and Davies had used. Surgically implanted osmotic mini pumps through a stainless steel gauge 25 cannula into the right lateral ventricle, which Morris learned to do in Lynch's lab, to limit drug exposures exclusively to hippocampus. Key findings: on the transfer trial in the hidden platform on the water maze task, day 9, after 15 training trials, the DLAP5 infused animals were still swimming all over the water maze. There's the amount of time they spent in each quadrant. And the control that just got saline through theirs were spending the bulk of time after that platform was removed, swimming around in the quadrant where the training took place, and you can see the extent of time they spent in it compared to the other quadrants. And the LAP5, which is a less efficient form of the DLAP5, these animals were also spending the bulk of their time swimming around in the quadrant where the platform had been during training. Furthermore, the DLAP5 treated rats couldn't learn a reversal task on days 10 and 13 of testing, but both control groups could, where now you put the platform in a different quadrant and train the animals on that. But these animals could learn a visual platform task where there's visual cues on the two platforms, different visual cues. One of those platforms is attached to a sturdy base in the pool. The other is attached to a wobbly base in the pool. So the animal needs to learn that that visual cue is where I want to swim, because if I swim there, I'm going to fall off as soon as I get up on it. They were completely intact in this study compared to the two control groups. So DLAP5 is not a motivational or visual disruption, because the rats are motivated to get out of that tank, showing here, it's not a visual disruption. They can solve this task. It's a memory disruption. But it did affect hippocampus LTP induction, measured in vivo, via chronically implanted stimulating and recording electrodes. Okay, so here's what Morris says at the end of that paper. We have shown that AP5 impairs place but not visual discrimination learning, a profile similar, although not as severe as that caused by hippocampus lesions. The profile of impairment with AP5 points to a mechanism involving interruption of hippocampal N-methyl D-aspartate receptors. And after these results, we were even further on our way to our current model of NMDA activation underlying both LTP and various forms of memory. Okay, the mid-1980s to present day, these experiments to further our understanding of these receptors are all based on the impact of new research tools. I'm going to be very brief here, that there's great stuff to talk about here. So using then recently developed recombinant protein synthesis and purification techniques from biotechnology, Shigemoto's Kyoto lab reports first successful clone of the complementary DNA encoding the rat N-methyl D-aspartate receptor synthesis of the full protein that you could derive from the clonal DNA, the comparable physiological activities of wild-type DNA receptors with the synthesized protein, and the first molecular model of NMDA receptor protein structure based on nucleotide sequence comparisons with previously cloned and modeled AMPA receptors. 2003, what happens between the early 90s and the early 2000s? We get further advances in biotechnology that amplify the quantity of pure protein generated by recombinant techniques. And in 2003, a graduate student, Hiro Funikawa, and Eric Genau at Columbia reported the first X-ray crystallographic images of an NMDA receptor subunit bound to neurotransmitter glycine, showing numerous structure-function relationships in that single subunit. And 2 years later, the same lab reports the first X-ray crystallographic images of the two NMDA receptor subunits bound to both glycine and glutamate. Now, the next 10 years was a piecemeal attempt to try to derive enough of that protein to figure out other subunits in a kind of piecemeal approach. And the reason for why you had to adopt a piecemeal approach is because N-methyl D-aspartate receptor is a heterotetrameric protein. And no one could figure out how to get one of these through the process of recombinant techniques. Hirohito Funikawa succeeds. They're the first to succeed in figuring out how to get a heterotetrameric protein using recombinant techniques in enough quantity to where you could do your X-ray crystallography. And in a 2014 paper, they provided the first X-ray crystallographic images of the full NMDA receptor protein in closed configuration with comparison to previously imaged homotetrameric AMPA receptors. And they provided the description, now widely quoted in textbooks, quote, the glutamate, their particular version of the receptor, bound to glycine, L-glutamate, is shaped like a hot air balloon. And that's been quoted in many textbooks. There it is right there. Okay, now, 2 years later, published in Nature, out of the same lab, Funikawa's lab at Cold Spring Harbor, using basically the tetrameric protein that Caracas and Furukawa had developed. They are able then to, in another new research tool, single particle cryo-electron microscopy. Here's a photograph of a this is the one at Vanderbilt, not at Cold Spring Harbor. But basically, the review paper in Nature Methods about this kind of microscopy was published in December 2nd, 2015. So they were already using it before the first review paper came out. Very recent technological advances in sample preparation, computation, and especially instrumentation are now allowing researchers to use cryo-electron microscopy to solve near atomic resolution macromolecule structures. And that's what we get in this paper from 2 years later in Nature, not only our first look at the crystal structure of the NMDA receptor in active configuration, all right, but also with no allosteric terminal domain antagonist, and they were able to measure the rotation at the subunits relative to one another and the vertical movement of two particular residues in that amino acid to dilate the gating and which is likely to lead to ion channel opening. If you go look at this paper, they've got the coolest in the supplementary village. They actually made animations of that protein described at that amino acid level and animations of it opening and closing. And it's just awesome to watch. Okay, and so the evidence stands today on the structure function of the N-methyl D-aspartate receptor after 6 decades of identity, we call identity experiments. Now, what lessons about scientific practices should we draw from this case study? All right, first, many of the key experiments were not hypothetico-deductive, conjectural refutational, Bayesian conformational, or our Slab, Lemmerth, and Bickle connection experiments in their basic structure and design. Why not? For decades, not enough theory or hypotheses about NMDA respect to structure or function was in place to guide experimentation of that sort. These were just simple explorations of this particular type. So they definitely meet our account of identity experiments, our framework source experiments in molecular cellular cognition, according to their fundamental goals. Type 1 is to discover new phenomena and understand their properties, identity experiments. But here's the question I want to pose now. Are some of these experiments I just described to you exploratory experiments, described previously by philosophers of science who also emphasize that exploratory experiments are not theory or hypothesis conformational in design? Now, here I want to turn to a paper that has unfortunately been forgotten to some degree in the experimental, in the exploratory experimental philosophy of science literature. And this is a paper by Kevin Elliott, Varieties of Exploratory Experimentation in Nanotoxicology, published back in 2007. Because I think this paper still provides what I consider the broadest account of exploratory experiments in the published literature. And here is the way Elliott describes exploratory experiments. Does not aim to test theories or hypotheses, and 2, involves extensive variation of experimental parameters. And from that basic condition on what these experiments are, this suggests to Elliott a taxonomy that varies along 3 dimensions. The positive aim of the experimental activity, the role that theory plays in the experimental activity, and the methods or strategies used for varying parameters. And that's what he begins to explore using this example he draws from nanotoxicology. Now let me start with this first kind, the positive aim of the experimental activity. He gives us a number of kinds of aims that these exploratory experiments have that these hypothesis confirmation ones do not have. Defining new concepts and isolating or manipulating particular entities or phenomena to study their basic properties. And I think you can clearly see that these 2 experimental aims were prominent in that 60-year history of the development of the N-methyl D-aspartate receptor. The second condition is the role that theory plays in the experimental activity. For example, he gives some examples, playing a minimal role relative to other forms of experimentation, like hypothesis confirmation, and providing background information. Now, this idea about exploratory experiments, the limited role that theory plays in, has been a really important notion that's been developed in the exploratory experimental literature. Okay, it goes back to Laura Franklin's 2005 paper, where she distinguishes between experiments, theory directed by both theoretical background and local theory of the particular objects being measured, versus experiments directed only by the theoretical background. That's what the exploratory experiments are. And I think to even more work, Dick Burian and Ken Waters, both 2007 papers, distinguish between theory-driven experiments, distinguished from theory-informed experiments, based on the expectation of what the investigators will find. Theory does not provide this, it does not provide these kinds of expectations in exploratory experiments. And finally, my postdoc who was with me, David Colexo, wrote a really nice paper showing that local theories of the target phenomena can inform auxiliary hypotheses in exploratory experimentation rather than the hypothesis which is being confirmed or refuted. Okay, so there's those two conditions. And finally, there's the methods or strategies used for varying parameters that the Elliott lists, using multiple experimental techniques to characterize the phenomena and working as a community to design a range of experiments that vary these key parameters. And I think the 60-year history of these N-methyl D-aspartate experiments beautifully reflect multiple experimental techniques, working as a community of researchers of a variety of different fields in order to design a range of experiments that vary these key parameters, leading to discovery. Okay, now clearly, the kinds of experiments I illustrated for you appear to meet these very conditions. So they appear to be a kind of exploratory experimentation. Okay, but if they are, then they reveal at least 3 features of exploratory experimentation that have not been noticed in the published literature. First, the role of sheer serendipity. I mean, you saw numerous instances of it. The 500-gram bottle of sodium glutamate sitting on the Canberra lab shelf. The scientific specialties of the particular key researchers. Organic chemists, for glutamate's added, the key early researchers, and the initial, quote-unquote, discovery of magnesium effects at some glutamate transmitters. Those can only be described as serendipitous occurrences, and they were crucially important because basically all of these features of the experimental just set the stage for the next decade, 2 decades, even 3 decades of the kinds of experiments that were done. And that has not been stressed in the exploratory experimentation literature. Second, discoveries in adjacent research areas, in this case beyond structural biology and neurophysiology. You saw the work of the biochemists, the pharmacologists, the LTP researchers, the behavioral neuroscientists, the biotech specialists, the geneticists, all further discoveries of these key features. And finally, the third one that has not been, let me just say that I've got a really cool research project going on with Laura Grodowski here from Pitt HPS. She wrote a dissertation at CUNY on the role of outsiders in scientific discovery. And we've been developing a number of cases together. And I think some interesting stuff will be coming from that. And finally, the third feature that has not been discussed in the literature, the monumental impact of then new research tools quickly put to use in investigating the emerging phenomena. That's one that's near and dear to my heart, so I've been looking so much at tool development. The patch clamp, the calcium indicator dyes, the molecular cloning techniques, first for predicting protein structure from clonal DNA sequence, then for generating sufficient amounts of protein for X-ray crystallography studies, and of course the last one, the cryotransmission electron microscope. Now, I want to emphasize that this is not the use of exploratory experimentation to discover or justify new instrumentation. That has been discussed in the EE literature. Rather, it's the use of these brand new tools at the time to drive these experimental discoveries about the target phenomenon. And finally, the only EE discussion I know that emphasizes the use of research tools in this way is a recent philosophy of science paper by Philippe Haïs at Bielefeld. I was one of the paper's reviewers, and my views on research tool development is what drove Philippe to write that paper, though we do disagree about the respective roles of tools in theory. All of these features seem to be crucially important missing pieces to add to our developing account of exploratory experimentation in the philosophy of science. With that, I will say enough from me. I can't wait to hear what you say, and we'll just take this guy's advice. There are no stupid questions, so let's agree there's no stupid answer. Thanks. Okay. And I think people are going to come up here and ask questions. Wasn't that the idea? Okay. Please.
So how do I do this?
Yeah, I'm not sure of the details.
Talking to this? Or talking near it? Do you want me to
Okay.
I've never done this.
Okay.
First of all, thank you. That was
Sure.
awesome. I was struck by, at the very beginning of the talk, you talked about analogs. And it seemed to me that there was almost like a role of brute force kinds of attacks to these problems where you have a list and you just go until you hit the right one.
Yeah, you bet. Yeah.
Has that been really discussed in this exploratory
Not that I know about, you know. I mean, in the exploratory experimentation literature, one of the nice things about this case is that, you know, you had, and again, it wasn't so much brute force. I mean, there was a kind of theoretical background, organic chemistry, biochemistry, that were actually informing where to go next, okay? Now, but you weren't confirming or disconfirming any theory about how these things work. You were just saying, okay, the next potential antagonist to derive is this chemical because of its location in the biochemical sequences. And that's just the way Watkins moved through these, right? And he found, of course, eventually, he found more, there's some that just worked better than others. That's how he and Davies were just kind of wading their way through, just using his basic knowledge of biochemistry and using Davis's capacity to do the neurophysiology. Now, that I think is a kind of interaction, right? That I do think that that's been somewhat discussed in the exploratory experimental literature with their discussion of the distinction between the amount of theory used in confirmation experiments versus the amount of theory used in exploratory experiments. So I think that kind of touches on this issue, because clearly there was theory in the background that was driving, that was informing these experiments. But these experiments were not designed, and they were not designed to confirm or disconfirm a hypothesis. Great.
So thank you for the talk.
Sure.
So I'm not really
Okay.
maybe closer.
Yeah, there you go.
So I'm not very acute to philosophy of science.
Yeah.
I'm a neuro.
Yeah, good. Yeah.
And so I'm wondering if it's about exploratory experiments is whether or not it's something that's more for when something like NMDA receptors first get discovered, because right now that we know more about like the mechanisms by which it goes, that's now every scientist that studies this receptor already has a theory that they do.
Yeah, I think you're right. I think there's a kind of dynamics of exploratory experimentation, and you're right. And one of the reasons you're doing exploratory experiments in these early days, you just don't have a theory to back. You got some theory in the background, sort of informing various directions you go, but you don't have a theory to confirm or to disconfirm. And that's, I think there is a kind of developmental dynamics at which exploratory experiments will play an important role. But again, I mean, it could also be, as I think we see as we kind of get a better and better picture emerging of how these things work and what they do, then there's still room for exploratory experimentation. I mean, it's not like it disappears. Now, I know that, you know, a lot of the funding sources now are insisting upon hypothesis confirmation experiments. But look, I mean, that's just an ignorance of the history of neuroscience. The exploratory experiments are hugely important, and there's still room for exploratory experiments, and there still ought to be funding sources for doing this kind of work. I'll get off my soapbox.
Adam and then Chris.
Great.
Yeah, thanks for the talk. That was really interesting. One thing, just kind of going off
good.
the end of your answer to that question, thinking about funding sources and maybe the motivations for why we're thinking about exploratory experiments. I wonder if there's maybe a reason that the role of serendipity is not talked about.
Yeah, good.
As a methodology, you don't want to be like...
I just hope you get lucky. Yeah, give me this money, because I really think I'm going to get lucky. Go to Vegas, you know.
I have a bunch of weird hobbies that might impact
Yeah, exactly.
So I guess I wonder, how important do you think that is? There's sort of two different thought, like a comment here, just on whether we're trying to describe the methodology as like a prescriptive way or a more descriptive historical way. I think I completely agree with you in the sort of
descriptive. Yeah, that's
as a prescriptive way. How important do you think?
No, great. Yeah, great. So look, and there's just so many examples from the history of neuroscience where just this serendipity, but it's serendipity in a particular way. It's serendipity that was noticed by really, really good bench scientists. I mean, take Hubel and Weasel's discovery of the vertical bars of light. You know, that first discovery, they were showing all kinds of elaborate shapes to the cats while they were sitting, and they're getting nothing. And then all of a sudden they drop one slide in and pop, 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 pop. And they go, what? What happened here? They go, drop it in again, pop, 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 pop. Pull it out. There's a vertical crack in the bottom of the slide. I mean, and oh man, and then now, and Hubel and Weasel, being astute bench scientists, man, maybe this thing's responding to bars of light in particular orientations, and they're off and running. Nobel Prize. Okay, so yeah, but now on this point, though, I think it's crucially important because I agree. We don't try to teach scientists to be lucky. Budding young scientists be lucky. But what you try to teach them to do is be attuned to unexpected results. Now that's a skill you can learn. And here's the way I'm trying to teach it to my honors undergraduate neuroscience at Mississippi State. I teach two versions of Introduction to Neuroscience to honor students. I teach one out of just the Purvis textbook because it's such a good textbook to use for undergraduates. The other, though, the next semester, what we do is we go back and read the original publications that first reported those landmark results. And I point out, here's the tools that were used and here's why. And you can actually see in a number of these cases that these were scientists that were serendipitous, but they were responding to the unexpected. The unexpected wasn't just immediately calling them to just trash that data. They were looking and saying, maybe there's something here. And that sort of persistence just has paid off in the history of neuroscience, and it will continue to pay off today. So I'm trying to teach budding young neuroscientists to be on the lookout for the unexpected, and maybe even set up some explorations of potentially unexpected results. Now, I'm not saying, what's the old line, blow a trumpet before the roses, and see how they grow. I'm not suggesting that. The tulips, right, exactly. And I'm not suggesting that, but I am suggesting an attunement to unexpected results that may lead to a major insight. And I think if we can train that in our younger scientists, we might get more that do this kind of discovery.
Yeah, cool. So, yes, it's more the response.
Yeah. Exactly. Yep, exactly. Great, thanks.
Chris?
Yeah. I had a very similar set of questions.
Yeah.
Just about the sort of normative dimension.
Yeah.
Of this. So imagine, as philosophers, a scientist comes in and says, okay, you've been thinking about exploratory experiments. What have you determined, and how is that going to help us do what we do? One kind of question would be, when is it that a field should be doing more exploratory work? And a different kind of question is, how do you do exploratory work well?
No, exactly. No, great question. But here's at least a strategy for trying to understand, especially your second question, which is actually crucially important. Articulating these kind of historical cases. And not just in terms of 4 paragraphs. I mean in terms of the explicit scientific detail that goes beyond the published work and actually digs into things like lab notebooks and actually digs into things like the vast supplementary material that's published to actually look and see what was the reasoning going on, what motivated these particular scientists to do that and to notice that. And I think that when you tell these kind of cases, in all this gory descriptive detail, it's for a reason. Because you want people to see, just kind of like how this kind of unrolled out with the idea that maybe maybe we can get other things to roll out in this particular way. Look, this is at bottom a descriptive account. Because that's what I like to tell these elaborate descriptive stories about experimental design, experimental practice, tinkering in the lab, all that kind of, that's what I like to tell. But I think that the motivation for doing, the prescriptive motivation for doing that is the hope that people will be more attuned to some of these events that you can kind of lay out and kind of just show how they ended up shaping this particular outcome.
And back to the first question. I'm curious what your views are about funding. So should there be fields where funding
Good.
Prioritize exploratory work?
That's an awesome question. And here's my thoughts about it. I mean, and so this is something. So Silva and Alcino and I are engaged right now in a discussion of just the impact of outsiders in science. Silva was a molecular geneticist at training. He got his PhD in human genetics at the University of Utah. He went to work in the lab of an immunologist, Asuna Tonegawa, 1 year after Tonegawa got the Nobel Prize for discovering the mechanisms of antibody diversity. And you know why Tonegawa wanted a neuroscientist, a potential brain study in his lab? He had taken a course at Cold Spring Harbor and thought, this is cool, I want to start doing this stuff. And Silva, comes to him and says, look, I just learned how, the techniques of knockout genetics. We can target any particular protein we want, knock out that gene, and then test these animals. And Tonegawa said, sure, come on, I'll give you some money to do that. And Tonegawa had, 50 postdocs working on immunology. This was amusing to him. It really is. That's what Alcino says. And so what happens here is that so Alcino and I have been talking about that. How can we get more outsiders to move over across disciplines to bring potentially new tools in? Because, so Silva basically knocks out the gene for the alpha isoform of calmodulin kinase 2, and 3 years later, there's 10 pages in Science describing the knockout, the physiology, and the behavior. And he shows how that affects behavior. And so there's and then within 10 years, virtually every behavioral neuroscience, every neurophysiology lab is using knockout mice and refinements of them. So here's what we're thinking. I mean, think of the humongous budget of and I'm just talking about America here, of the NIH. $47 billion last year, I think it was their research budget. Let's take 1% of that, okay, and let's use that to basically give people that that either have been extremely creative or have whatever we think it takes to do this kind of creativity work, give them a certain amount of money, don't have to write up annual reports, and just turn them loose. Now, I want to say 1% of the budget and just turn them loose. And then 5 years later, let's assess. I mean, it wouldn't be that expensive. Now, yeah, there's going to be some dumbass congressman saying, what are we spending money on this for? But that's just political reality. So, I mean, there are ways of thinking about doing this kind of fun. And I will say this, there is some attempt to do this. The BRAIN initiative, the federal and the corporate initiative in the states, which is trying to develop new technologies to do neuroscience, has been hugely successful. And they've been basically taking an approach to where you just convince them that there's some promise in the tool you're trying to develop, and they basically do this. They basically give you this money, and you have at it. So there's some movement afoot, it seems to me, in neuroscience that's looking to do this kind of work. But, how much do we want? This is a hugely difficult question. How much do we want to dedicate to exploration? And that's different people are going to have different intuitions on that. Yeah.
Mike and Julie, Mike had his hand up before you. You can stay, and then.
So I just wanted to basically make a comment on the answer to your previous thing, which is that I think not just attention to the cases where people successfully pay attention to the unexpected, but where they didn't.
Missed. Right? Yeah, where they missed.
So the one that comes to mind first is Penfield's motor hormone. So in fact, Penfield saw the behaviors and gathered the data that underlie the current understanding of the organization.
Cool.
But because he was theory driven,
good.
He missed it.
Yeah, good. That's awesome. Because here's Calexo, when he was my postdoc, here's what he asked me. He said, John, you've been writing about all these remarkably successful labs. You've been studying the practices of these remarkably successful labs. Okay, Silva's lab, the work I did, was Hubel and Wiesel's work, and Hodgkin and Huxley's wet lab work. I mean, it's all this kind of history I've been doing. These remarkably successful labs. Do you think you're actually biasing your descriptive account of science? Most labs aren't that successful. don't you think you need to go into these? Hey, Joan, your lab's pretty mediocre. Can I come study what you do? I mean, you know, how do you do it? I don't know. But it's a good point. I mean, you know, it's a good point. I mean, like, because we may be presenting a skewed picture of science here by understanding how these spectacularly successful labs have gone about doing things, if we're interested in giving a descriptive account of science. Now, I don't want because like, look, there's a huge amount of work that's necessary for labs that are just taking some new, hot, great development and building it up. I mean, you know, we need the pioneers of the science, and we need the settlers of science that just go take those ideas and build them into what we have today, and make new drugs and and basically flesh out the account. So there's a give and take here. But, you know, I do think that we underfund pioneering efforts in the sciences, especially in the neurosciences. And it would be nice to see if there was sort of a more emphasis. But again, I agree. I mean, what is the method of scientific discovery? Well, there isn't one. Here's a case where we can show you how that developed. Or we can show you, for example, how the patch clamp was developed from 1972 to 1981, and how the gigacil was discovered, and guess what? It was by luck. And it just so happened that Sackman and Neher just happened to be lab mates with each other. Neher had just done a dissertation in Munich on how to basically develop glass micropipettes. Sackman had been working in Katz's lab, was the world's expert on how to polish membrane. And they just happened to find each other next to each other in a Max Planck. And hey, why don't we combine our talents? And 10 years later, the patch clamp. Yeah, that's just kind of, but you got to kind of go to the gory details to lay out how that works, to see if there's anything we can learn from these cases that generalizes beyond the idiosyncrasies of the particular case. Great. Oh, great.
Hey, so I guess I'm kind of bridging.
Yeah, good, cool. Yeah.
Questions.
Yeah.
And I'm taking the perspective of a trainee here.
Okay, good.
But, so I was wondering, and I don't know how you would even get this data.
Yeah.
I'm wondering if there's anything out there that compares the amount of time it takes to publish.
Yeah.
Like an exploratory experiment versus another one.
Yeah.
And what proportion fail.
Yeah.
Because we've talked about the issue with funding.
Yes.
Another issue, obviously.
Yeah.
In science for trainees is the whole
you bet.
Perish.
You bet. Yep.
So it's one thing for people like Tanagawa or John O'Keefe to go
exactly.
in the lab after hours.
Right, exactly. No, you're right. You're exactly right. I mean, so there's that aspect of science, again, this aspect of institutional science where we all know how long publication takes. Right, but now, of course, we can get these pre-pubs and stuff like that. All that stuff can be put on. But nevertheless, there's these institutional barriers to doing exploratory experimentation because you're sinking a potential career, and if you fail, your career's over. I mean, so if you sink 5 years into discovering some new fact, and you don't discover it, it's tough luck. I mean, there's this, but how about if we did this? How about if we kind of widely came to recognize among funding sources that there are these two important aspects of science. There's discovery, and then there's justification and kind of fleshing out the details. Both are absolutely crucial to scientific practice. And certain people are better at doing certain of these things, and certain people have personalities that just kind of fit better with these different kinds of approaches. And so how if all graduate students came to appreciate the two aspects here of science, the creative discovery aspect and the confirmation development aspect, and you just choose which path looks better for you? And would you be happier doing this kind of work or this kind of work? And if that was just kind of somehow institutionalized, maybe we could actually get those people that have either a desire or a knack for creativity to give it a shot And then those who are just, I want to just kind of develop, okay, we've gotten this far with this receptor. I'm just going to flesh out one more detail of it. And they could be perfectly happy doing their kind of science. They could both get funding from different sources. And who knows what would happen. But it seems like it's going to be better than where we are now.
But do you actually think that those two streams could really exist? It almost seems to me like when the serendipity happens, sometimes it's in the theory, like you're saying you train your neuroscience students to be attuned to odd data.
Yeah. Because I show them where that is in that. So we begin by reading a Penfield paper because everybody finds Penfield stuff so cool, the microstimulation. So that gets them off the ground, okay? And then what we basically work our way through is that we do a little bit of the stuff of the kind of the early neuropsychology of memory work. We read Scoville and Milner. We read Corkin's early paper with HM. We read a couple of papers from the Squire lab, from the 80s. And we just kind of work our way through the neuropsychology of memory. Then we dig in on then they read a Kandel and Hawkins paper, aplesia, and so we work on that, and then we move to the developmental history of LTP. And so they read Bliss and Lomo. They then read Collingridge. They then read Lynch. And then they read the Lynch-Morris paper, and then they read the Silva-Tonegawa papers with the gene. And every step of the way, you're going, look, what was novel here? What was new here? What insight did these people have, or what freak event did these people see that enabled them to do this piece of research? And we're reading the original publications. So of course, now we all know that scientific publications are not a picture of the day-to-day lab work that led to them. They're highly refined pieces of writing, okay? But if you know enough about the history of that detail, you can tell people, they didn't say this in the paper, but guess what? This is what happened. And just to get them to be attuned to, and look, some people it resonates with, some people it doesn't, right? Some people are just sort of born explorers, others are born builders, and great. Let's exploit that fact about human personalities and try to direct people in one direction or the other, depending on their personality, depending on their skills, depending on what they want out of a scientific career. I mean, it'll be better than we are now, where it's just a free-for-all, and you got to tell a grant agency that, if this works, I might cure 10% of deafness. But really, I just want to figure this out.
Thanks, John. Just as an exploratory question.
Good.
What more can philosophers learn from the spirit of discovery in science? Can philosophers of science probably be explorers themselves? So is there something that philosophers of science can learn from the story?
That's a great. That's what I'm really. That's a great question, right? Because that's why I said here's the philosophical lessons I see in this. What do you see? I mean, maybe there's more. Maybe there's something I'm just flat out missing. Okay, that's a really interesting consequences for the way we think about how science works. So that's why I like to tell these kind of detailed stories about how these experiments were done, what tools were used, why those experimental choices were made, how that design choice kind of survived and got tinkered with in day-to-day lab work. I like to just dig in and tell those stories because the question is, what are we learning significant about how science works by investigating these kinds of stories? Now, I am completely convinced, however, that you're only going to answer the question, how does science work, by studying scientific practice. You are not going to learn that by studying long and hard at scientific products. You got to study how it works in the lab on a day-to-day basis, how these choices are made, how these designs are made, if you want to understand how science works. You're not going to learn that by studying, what is an explanation in science? because that's not how explanations kind of emerge from these practices. And do they have a logic? Well, I don't know, But I want to know how science works so I can think of when science has been doing well and when it's not been doing well. And to me, studying practice is the only way to do that. Studying scientific practice.
And the push is for philosophy students to go visit these labs?
You bet. You bet. If you're going to come work with me, as a pre-doc or a postdoc, we're going to get you in a lab if you've never been there. Well, I'm going to take you down to my colleague, my collaborator, Brad Walter's lab. Brad's a molecular geneticist that turned to neuroscience during his postdoc at Children's Hospital in Memphis, St. Jude's Hospital in Memphis. And now he's trying to cook up the right combination of kind of genetic material that you can vector into these support cells in the inner ear and turn them into active hair cells. And you talk about, I mean, you're going to learn microscopy. You're going to learn, you're not yet going to learn physiology because you haven't got to the point where you can test. But you're going to learn how to do, just simply viral vectors to insert genetic material, because this isn't an adult rodent model. This is not in stem cells or anything like that. These cells have already differentiated. What can we do to make them redifferentiate into it? That's what he's working on. It's fascinating work. And I'll take you down there and you'll spend six months, just seeing how that kind of lab work works. Because I think that that's the only way that you're going to come to a philosophical understanding of how science works.
We do have a lab associates program for
And it's no, it's great. And I mean, Jackie, you were one of the first, so I was one of the first people. You were kind of the next people to do you got a master's in neuroscience. You spent a lot of time in the lab. So, and and that's what led you to make a lot of your important early papers.
No.
I mean, the 2009 paper that's been cited so much basically came out of the fact that you were embedded in a lab doing the work and seeing how it actually worked. So, and I think that's become more and more prevalent in philosophy of science nowadays, certainly in philosophy of neuroscience nowadays. So pretty much every young philosopher of neuroscience I know has spent some time in a lab. I mean, they may not have gotten a master's or spent 3 years in it, but they've spent some time in a lab to see how this stuff works.
No, I'm just saying we're sympathetic to that.
Yeah, you bet.
We've been trying to do it here for a while.
Exactly. Exactly. You bet.
For years.
Yep. Yep.
Yeah. Yeah, somebody else had their hand. Okay,
Great. Yep. Awesome.
So this is also kind of a normative question.
Sure.
And it's about funding. And you may have already answered bits of this before, but I was wondering if you could make it explicit. A little bit more explicit.
Yeah.
Let's say I work for the NIH.
And
there has been this 1% of the budget. That's all done. I have 1,000 grant applications for exploratory research. How do I choose who gets them?
Well, first of all, you want to, okay, look, there's a bigger problem here too. But you want to do, is there any inclination that this particular applicant has that kind of creative bent? Do they have any kind of history at all of that? But the bigger problem is this. Suppose you are that NIH director and you've got this money, you're going to kick it to 5 years for discovery, and what if 5 years later, 0 discoveries have been made? Guess what? You're at the door. I mean, you're gone. I mean, that was your project. That was your, that was your baby, and it failed, and see you later. Go back to wherever. So and so there's a lot. There's these dual motivations to fund what's going to be successful. Okay, and how can you, in any sense, predict successful discovery? You can't. Okay, but you can rely on some features of those kind of people that like to do this kind of discovery. They have a certain type of personality. They have this, and maybe they've shown these sort of aspects of creative discovery in some of their prior work. Or maybe the person that was their PI can say, hey, this person really likes kind of like trying to cook up new ideas. And I can go, okay, cool. This is kind of person we want to try to train in doing this kind of work. So, I mean, it's not like there's no algorithm for discovery. We all know that. But there's certain kind of personality features and work features that the really good discoverers display. Do we find that in younger scientists? And if we do we develop that in younger scientists and try then to give them the resources to do that kind of work? That's what I'm trying to convince what we need to do by just looking at the history of neuroscience. And not just glancing at the history, but actually digging into and finding out what were the steps that produced this result. That's the hope. That's the hope.
Yeah, I think what you're talking about, John, is importing a certain venture capital.
In a sense, that's exactly right. In a sense, that's exactly right. And there are biotechnology firms that do that kind of stuff, right? And there's pharmacology firms that do that kind of stuff. But could it be more institutionalized in scientific practice, to where it was widely recognized as something that we're trying to cultivate in science? I don't know. I'd like to say yes, but on the basis of this, but yeah. Oh, yeah, sorry.
Okay, so I have two questions, but
sure.
I'll just try to keep them short. So
sure,
the first one is, what does this historical case mean, or does it have any force or influence on debates about the value of data-driven research, discovery, science in exploratory experimentation?
Yeah.
Do you think it has any force in that debate?
Yeah.
And then the second question, kind of connected, is this demonstrative of how exploratory experiments can be standardized?
Good question. And the answer to that second question, I have no idea. But let's do some more of these and find out. Let's do some more of these kind of relatively deep, detailed dives into these kinds of studies, right, into this kind of work, and see if we find commonalities across them. I don't know if we will, but we might. That's why, as I've said before, when Sylvan Landreth and I wrote that book at the, on the basically the experimental design practices in molecular and cellular cognition, at the end of that book we said, okay, does this generalize across other sciences? We don't know. But sure it'd be cool if a systems neuroscientist who's also interested in these kind of broader issues did this kind of meta-scientific analysis of systems neuroscience. And then would have that in place, and then let's have someone that works in computational neuroscience do theirs, and then we can cross-check. Are the experimental design practices similar or different? Where do they differ? And I think the similar kind of thing here with these kind of processes of discoveries. Let's just work out a bunch of these detailed cases and then compare and contrast. How is this case like that case? How is that case like that? And just see if any kind of this is exploratory philosophy, right? Here's a case right here. What a cool case. What an important case, okay? What can we generalize from this case? And that's going to be we kind of have to do more of these studies of these kinds of cases and find out if there's any general themes at all. I suspect there will be. Yeah. But I could be wrong.
Mel?
I have a question.
Sure. Oh, great, fantastic. Come on. Great, awesome. Great. Excellent, Mel.
One thing that universities around the world are doing is setting up institutes where they bring together people from different disciplines.
Yes.
Have you begun, or any of your colleagues begun to explore what role they may play
in discovery? Yeah, no, that's a fascinating question, and the answer to that question is no, but what a great project to look at. Let's take a look at one of these, or two or three of these institutions that have been successful in this regard, and let's find out what they do. And let's find out how they interact. And let's find out what the kind of day-to-day interactive practices are. Because the big problem, of course, is that you get these kind of multidisciplinary institutes, and the chemists do their thing, and the physiologists do their thing, and they kind of meet and talk with each other, and maybe one will kind of like collaborate with another, but really they do their operation as in their particular discipline. But let's take a look at let's find some examples of institutions like the ones you're describing, that have really generated something that was really influential and really kind of stellar. And let's find out how they did it.
It would be interesting to compare
Yes.
those that have websites only.
Interesting, yep.
Those that have Ubiquity working.
Yep, you bet.
People are actually thrust into the same building.
Yes.
And they have fun together.
And
I don't know who you're thinking of.
Yes. No, you're exactly right. You're exactly right. No, that's exactly right. And let's just kind of figure out from these detailed case studies a set of best practices that someone could implement if they were going to start one of these new institutes. Now, of course, we got one huge problem, and that was the last 3 years. Okay, because normal human interaction ceased. Normal human interaction ceased during those 3 years in any kind of real sense. And so we're going to have a gap. And we're going to just see, but here, let's do this. Let's see how these institutions recover, which ones recover better and most quickly, and figure out what they're doing. Okay, and then try to use that for normative payoff.
You also, straightforwardly, you could just look at productivity measures in the 3 years pre-COVID.
Yeah.
And the 3 years since.
Yeah, exactly.
And then compare institutions within and between.
Yep, and then go try to figure out what those successful what those successful ones did. What did they do? What did they do? Now again, you might find all kinds of just weird, weird little quirks that just sort of happened. Okay, but again, that's important too, right? Because you want to be attuned to what you want to be attuned to, as I'm calling it, serendipity. You want to be attuned to the unexpected that has had a track record of productivity.
What if we take Bill and then you get to have the last word, Jonathan? Does that
Yeah, great. Well, this isn't even the last word. I mean, this is going to go on.
I'm not saying
exactly in this setting, and then we're going to move to another setting and keep this going.
One of the things about the historical story, it would be really useful. I mean, you could see really interesting cases of finding new phenomena, and then let people find connections to explain and illuminate.
Yeah.
It might be useful to look at in that long story, the cases where the new phenomena didn't get followed up very well.
Yeah, good.
And then look at what sorts of
good.
changes there are.
Yeah, good. No, that's an excellent point. And again, it goes back to this point. The big problem with looking at these historical cases this closely is you're generally looking at humongous successes. Right, the N-methyl D-aspartate receptor. I mean, one of the great discoveries and developments in late 20th, early 21st century neuroscience. I mean, there's just no two ways about it. But again, you got to wonder if you're kind of skewing your descriptive account when you just focus on these remarkably successful cases. So what if we look at periods of time where that development stalled, okay? Because there were periods of time, I kind of ran you through the basic chunks of time and what was happening in that chunk of time. But look, there were periods of years where just no progress was made. What was going on? People didn't stop doing the science, right? They kept doing the science. What were they doing wrong? What were they doing differently than they did? And knowing that story could be hugely normative for how we go about deciding how to institutionalize this. Yeah, so it goes back to this point about, you can look at the hugely successful cases, and that's important, but you probably ought to get a picture also of why the unsuccessful cases were unsuccessful. Yep, and I think that's a huge part of the story.
Jonathan, do you want to come up or you?
Sure. So I had a question about one of the early steps.
Okay, cool.
And I don't even have the vocabulary right
That's
now.
That's fine.
But I'll try.
Yeah.
So it's the part where they discovered an effect of magnesium ions.
Yeah, exactly.
But they weren't looking for that.
No,
So that's
exactly.
what they discovered.
Yeah.
But how did they notice that that's what was making the
Good.
difference?
Yeah, how do they notice? So here's what they're doing, okay? So they're basically just setting up their screening studies. Right, so okay, we're going to use frog spinal cord because Watkins had been parts of groups that had used that going back to the late 50s in Eccles lab, and we're going to do the same measurements that we've been doing to affect whether or not that set of receptors is responding to these different elements. Okay, but here's something we need. We need a tool that will enable us to just shut down neurotransmitter release at any given time just by injecting that particular substance into the frog ringer solution. So what are we going to choose? Well, we need a positive ion to shut off the presynaptic neurotransmitter release. And frog, here's one positive ion that frog ringer solution is bereft of, doesn't have any magnesium. So let's just choose that first. Let's just dump magnesium in there and see if it does what we want it to do, which basically just is a tool to stopping neurotransmission. Oh my God. It's halting the magnesium alone is shutting down when we add N-methyl D-aspartate or N-methyl L-aspartate. There's no response. When we add glutamate, we get this kind of intermediate shutting down. And when we add quisquelate and kainate, it's just going on as normal. Weird. There must be something about these receptors when we came to talk about N-methyl D-aspartate receptors that is responding to that magnesium in a way that the kainate and the quisquelate receptors, the AMPA receptors and kainate receptors, are not doing. And so it was just, again, sheer serendipity of the fact that we needed a positive ion to shut down neurotransmitter release, and we're going to use this one because frog ringer solution has none of it in it. That was that. It was that serendipitous.
Okay. So it was,
but they were cool. They were observant enough to see that this had only selective effects on these different antagonists of these particular glutamate receptors.
Okay, thanks.
Yeah. No. So yeah. So.
I apologize. I think Greg's.
Oh, sorry.
So I'm wondering if maybe we should thank John here and continue the discussion.
Yeah, let's continue the discussion. Thanks so much.